Hello and welcome to another video. Today we're going to be going through the majority of the topic of enzymes catered to the OCR A-level biology specification, but the video has been edited such that it is pretty much applicable to all major specifications. Now, when talking about enzymes, it's important we first understand what is an enzyme, what does it do, and where does it act. So, an enzyme essentially catalyzes a reaction and therefore increases the rate of that given reaction. The way it achieves this is by lowering the activation energy, as we'll go into more later. And enzymes do that on biological reactions, and they're biological catalysts. They are globular proteins. More on that can be seen if we think back to biological molecules as a topic. And they're also vital for controlling the majority of metabolic pathways. And they do this through an enzyme cascade, which is also again seen in more depth in later topics. Now, enzymes can act to catalyze a reaction intracellularly or extracellularly. So, an intracellular example is catalase. So, now when we talk about an enzyme working intracellularly, we're saying that it catalyzes reactions that go on inside of the cell. So, catalase is key to maintaining the pH balance in cells. It converts hydrogen peroxide into water and oxygen, which prevents excessive damage to cells. Now, the hydrogen peroxide arises because it's actually quite a common byproduct in a lot of metabolic reactions. Extracellular enzymes we can think of are mainly digestive enzymes. So, amylase is an example, which is key for the digestion of carbohydrates as a macromolecule. It will hydrolyze starch into simple sugars like maltose. Trypsin is another example, which is a digestive enzyme that aids the digestion of proteins into peptides. The key takeaway from this slide is that enzymes are biological catalysts, they are proteins, and they are vital to life as they regulate the majority of metabolic pathways and can act intracellularly and extracellularly. The examples I mentioned are good to know, but you don't necessarily have to know them. So now on to the actual method of enzyme action. So how do enzymes function? So every enzyme will have what's called an active site. This is a uniquely shaped or specifically shaped part of an enzyme which has the ability to bind to substrate molecules. So obviously an enzyme is a protein, therefore it has a specific tertiary structure, and therefore the active site's specific tertiary structure leads to a specific shape that is complementary to only the enzyme's specific substrate molecules. Certain conditions, which we'll get onto later, can change the shape of the active site and make it non-complementary to the substrate molecules. And when these factors affect the shape of the active site, an enzyme can be denatured, which means that it's no longer able to function. An enzyme essentially functions by providing an alternative reaction pathway with a lower activation energy. For those that don't know, activation energy is the energy required for a successful reaction to occur. So the outline mechanism of enzyme function will be as follows. A substrate binds to the active site. We see that in this first picture with the substrate molecule binding into that active site. An enzyme substrate complex will then be formed. A reaction occurs with the low activation energy and an enzyme product complex is formed. Then the products are released and the enzyme can work again. That's why it's a biological catalyst, because it's not, and again, don't use this in your exams, but the enzyme is not used up in any way during this, so it can repeat this process. So once the products are released, a new substrate will come and use that same enzyme, and that will happen however many times are required. Now, the reactions can be anabolic or catabolic. Catabolic includes the breakdown of complex molecules into simpler molecules with a single substrate and multiple products. So, hydrolysis, for example. Anabolic is when more complex molecules are formed from simpler ones. So, multiple substrates to form one product. So, you may be wondering how exactly the substrate will bind to the active site. Now, there have been a couple of theories surrounding this idea. So, the first was suggested by Fischer, and that was the lock and key hypothesis. So this indicates that the enzymes have a very specific active site and are rigid structures, so they are set in that mold that they have, and then the substrate molecule will just simply fit into that active site shape. The induced fit hypothesis is something that has been brought about more recently and is an adaptation of the lock and key hypothesis. So this indicates that the enzyme's active site can change shape very minimally. So when the bonds are made between the substrate molecule and the active site, so hydrogen bonding, disulfide links, etc., there are slight changes that occur to the active site to make it to make the bond as complementary as possible, and thus optimize enzyme activity and efficacy. So now we can move on to factors that affect enzyme activity. So there are four core ones that you need to know for A level. Those are pH, temperature, enzyme concentration, and substrate concentration. And we'll go over those in the next slides. So enzymes work within a narrow pH range and will work most effectively at their optimum pH, which is illustrated by the peak on the graph. This will typically be around neutral, which is obviously a pH of 7. A pH that's too high or too low alters the rate of reaction. When pH rises above 7 and becomes too high, an enzyme will begin to denature, meaning no, or at least far fewer enzyme substrate complexes can be formed, and in turn fewer enzyme product complexes are formed. Now, there are hydrogen bonds within the tertiary structure of a protein, and thus when there's more H plus ions in a more acidic environment, and obviously H plus ions are essentially protons, they interfere with the hydrogen bonds and also migrate to the negatively charged regions where ionic interactions are occurring in the tertiary structure of the enzyme and the active site. When the ionic interactions of an active site are altered, and this obviously occurs because the H plus ions will cluster to any negatively charged regions, the active site shape alters, and this can make it no longer complementary to the substrate molecules. The H plus ions especially replace the hydrogen bonds, and in doing so alter the active site shape. Now, different enzymes have different functions and act in different parts of the body, and are thus in different environments with different pHs. Enzymes have thus had to adapt to the pH surrounding them, and this has had an effect on the optimal pHs of given enzymes. So if we look at the example of the stomach, pepsin works in the stomach, and the stomach is, of course, a very acidic environment with a very low pH. So due to this, pepsin as an enzyme works best in more acidic pHs of 1 or 2, whereas proteins, which may work extracellularly, will have more neutral pHs of close to 7. And then amylase, which digests starch to maltose, will work optimally at about 6.8, which is far nearer to neutral as they're active in the oral cavity, which is obviously extracellular. So the amount of detail you need to know on how exactly pH affects the shape of the active site and enzyme function varies based on specification, but essentially all you need to know is, as mentioned, protons and H plus ions will cluster around the negatively charged areas of the amino acids in the protein, and any negatively charged R groups on the amino acid, which are exposed to form hydrogen bonds or ionic bonds, will have a group of H plus ions around them, and then that clustering alters the protein shape, as the H plus ions will take the place essentially of the hydrogen bonds, and what this leads to is the alpha helix of an enzyme to kind of stretch out more and kind of uncondense, so to speak. It's also important to note that pH changes to protein structure and active site and the active site are temporary unless at kind of extreme pHs, in which case the bonds may not be able to reform even when neutral pH or optimal pH is like reinstated. So the next factor that affects the rate of enzyme activity is temperature. So we can start by explaining the graph and the shape of the graph. As temperature increases, more kinetic energy is supplied to the molecules involved, and this thus leads to a faster molecular movement, and there are more successful collisions that occur between the enzyme and the substrate molecules. This means that there's more enzyme substrate complexes formed per second, and in turn more enzyme product complexes formed. This overall increases the speed of the reaction. We can see that this trend continues up until a point, known as the maximum point. After this maximum point, we see an acute drop in the rate of enzyme activity. And now this is essentially as a result of enzymes denaturing. So we can explain the effect of temperature on the enzyme as follows. So as the temperature increases, the hydrogen bonds within the protein are broken after that maximum point, especially as they do not require obviously a huge amount of energy to be broken. Now, it's important to note that the peptide bonds between nucleotides in the polypeptide chain aren't broken, as the energy required to overcome those bonds is considerably greater than the energy supplied by an excess of heat, and thus the primary structure of the protein is not altered at all. But due to the hydrogen bonds being broken, the protein is no longer in a tight, complex structure, and thus the hydrophobic, hydrophilic interactions and the other bonds in the tertiary structure are no longer acting either. The disulfide linkages, for example, between sulfur groups are broken too, and as such, the protein loses its shape, and thus the active site shape has changed to a point where it's no longer complementary with the substrate molecules, and thus the rate of the reaction decreases. It's also important that we understand what Q10 is. So Q10 is a temperature coefficient, which is calculated by dividing the rate of a given reaction at its higher temperature versus the rate of reaction at a lower temperature. Higher temperature is usually the same temperature plus 10 degrees in degrees Celsius. Now, it's important in calculating the amount by which the rate of the reaction increases as the temperature is increased by 10 degrees. And the value in lab testing is used to determine the exact temperature increase required for optimal reaction rate and kind of identifying when an enzyme will denature, etc. So now on to enzyme concentration and how enzyme concentration affects the rate of reaction. So as enzyme concentration increases, more active sites are essentially available, and thus more successful collisions occur, and so more enzyme substrate complexes can form, which in turn means more enzyme product complexes can form, and so more product is produced. That's obviously observed on the graph because there's an increase in the rate of reaction and enzyme activity as enzyme concentration is increased. During this time, the enzyme concentration is a limiting factor because it's what determines rate of reaction. The reaction rate eventually reaches a plateau, at which even if more enzymes were to be added, the rate of reaction would not increase any further. And at this stage, the substrate concentration is the limiting factor. Now, the plateau essentially occurs because all substrate molecules are occupied in enzyme substrate complexes, so they're all currently essentially reacting, if that makes sense. So adding more enzymes wouldn't do anything because there aren't any more substrate molecules to react, essentially. And so the reaction rate levels off, if that makes sense. Now, the dotted green line we see is a theoretical line if substrate concentration was continually increased. So if substrate concentration were, say, infinite, then as enzyme concentration increased, the rate of reaction would increase too. But obviously, in reality, substrate concentration isn't infinite. So what actually happens is a plateau forms, as we've just explained. Now, the next factor we can discuss is substrate concentration. Now, this is extremely similar to enzyme concentration, but I'll go over it for the sake of getting in all the points in essence. So the graph shows that as substrate concentration increases, the rate of enzyme activity and thus the rate of reaction increases too, up until a point which is the maximal rate at which a plateau is observed. And the enzyme concentration becomes the limiting factor or reagent, and thus the enzyme activity rate cannot further increase. The reason for the activity increasing as further substrate concentration is increased or more substrate is added is that more enzyme substrate complexes can form simply because there are more molecules present, which increases the likelihood and chance of a successful collision occurring. As a result, more enzyme substrate complexes form and more enzyme product complexes form, and thus more product is formed. At the maximum rate, adding further substrate will not lead to an increase in the rate of reaction, as nearly all the enzymes are occupied with substrates. Not even nearly all the enzymes are occupied. with substrates, and thus no successful collision could occur. Now you notice that each time I've explained one of these factors, I keep saying the enzyme substrate complexes, enzyme product complexes, etc., and all those key words. And now the reason I'm saying them in full is primarily because I found that, at least in my experience doing OCR, there are often marks just for those key words. And even if not, it's a good habit just to make sure you're going over the process in full, and at least know exactly what's happening. And obviously, right now, yes, I'm explaining all of them, but it's very rare that you're going to get a question that talks about all factors. But you may get a question that goes into detail about substrate concentration or enzyme concentration or temperature. And then in that case, it's very important to kind of fit in all your keywords and just try and maximize marks wherever possible. So that's the main reason I keep saying the full terms. Okay, so now on to coenzymes, cofactors, and prosthetic groups. Now, everything for these three is more or less written on the screen. So, coenzymes are organic compounds, organic cofactors, which bind non permanently to facilitate an enzyme's catalytic activity. They act to aid the bonding of the substrate to the enzyme. And then cofactors are non-protein compounds which are required for enzyme activity. They can be inorganic metal ions which bind temporarily and alter the active site to make the binding of the substrate to the active site possible, or more likely, or just simply aid the binding process. Now, prosthetic groups are permanently attached to the enzyme and aid the enzyme functioning in its in completing its function. So a prosthetic group would be a non-polypeptide group, and an example of this, which you'll almost definitely know, is heme and hemoglobin. All three of these should have come up in biological molecules, but if not, when I do the biological molecules episode, I will talk potentially a bit more about these. But for now, that's pretty much all you need to know. And yeah. Okay, so now on to inhibitors. So we have competitive inhibitors, which are enzyme inhibitors with a similar shape to that of the substrate molecule, and thus they compete with the substrate molecule to bond to the enzyme's active site. Once they successfully bond to an enzyme, it will block the active site and prevent further enzyme substrate complexes being formed on that enzyme. The degree of inhibition is based on the relative concentrations of the substrate molecule and of the inhibitor molecules. So, increased inhibitor molecule concentration means more successful inhibitor enzyme collisions per unit time, and therefore there's a decrease in the number of available active sites on enzymes, meaning fewer successful collisions between enzyme and substrate molecules, and therefore the rate of reaction will decrease. Now, if you increase substrate concentration, the effect of the inhibitor will be reduced, so there would be less successful enzyme inhibitor collisions, and thus the inhibition rate would be lower, and more enzyme product complexes, and so more enzyme substrate complexes, and thus enzyme product complexes would be able to form. Now, it's important to note that enzyme inhibitor complexes are catalytically inactive, even though they're called a complex, and an inhibitor bound to an enzyme won't be chemically changed once bound, in the same way that a substrate molecule would be, because there's no essential reaction happening. So if a competitive inhibitor binds irreversibly to an enzyme's active site, it's called an inactivator, but not all competitive inhibitors are inactivators, and most bind reversibly. It should also be noted that an inhibitor may not be fully complementary to an active site, so it might not necessarily be the exact same shape as a substrate molecule. It will just be complementary enough such that it can bind with the active site and essentially block it, if that makes sense. So the other type of inhibitor is a non-competitive inhibitor. So a non-competitive inhibitor binds to an enzyme at a point other than the active site. So that site is called an allosteric site on an enzyme. When this occurs, the tertiary structure of the protein is altered, which leads to a change in the shape of the active site, making it no longer complementary to the substrate molecule. Now, this will obviously inhibit the reaction, as no enzyme substrate complexes will be able to form. Now, a non-competitive enzyme inhibitor is not affected by an increase in substrate concentration, This is because no substrate molecule is complementary to the active site shape, and the inhibitor is not competing with the substrate molecule. It can bind regardless of substrate quantity, and therefore, and also once bound, no successful collision between the enzymes and the substrate can occur. Once all enzymes allosteric sites are inhibited, the reaction will essentially stop. And again, while some non-competitive inhibitors are reversibly bound, others can be irreversibly bound and just stop that enzyme altogether. So there's also end product inhibition, and this is a form of inhibition in which an enzyme functions as normal and catalyzes the production of product molecules, but this product molecule or molecules remains tightly bound to the enzyme in such a way that no further substrate can bind to the enzyme's active site. This is quite common in negative feedback loops, and it essentially plays the role of assuring a cell does not produce an excess of product and only produces as much as the cell requires. Essentially, the role that inhibitors play and end product inhibition plays is in regulating and controlling metabolic processes to ensure that the amount of product and the concentration of product we have in our body remains constant and in an optimal region. Now, it's important to know how inhibitors can function as poisons and kind of medicinal drugs. So, certain inhibitors are poisons. So cyanide, for example, is a non-reversible inhibitor of the enzyme cytochrome oxidase, which catalyzes as part of an aerobic respiration. So this sort of inhibitor is fatal, as the only way to reverse such an inhibition would be with new enzymes, which would take time to be produced. Cyanide is a type of metabolic poison, essentially, as it prevents a metabolic reaction from occurring properly. And metabolic poisons can be used in research to better understand enzyme function in certain reactions. Then we move on to medicinal drug inhibitors. So some inhibitors are positive in that they inhibit enzymes which would otherwise have negative consequences. So bacteria, for example, is inhibited by the inhibitor of penicillin in a non-reversible inhibition. Aspirin non-reversibly inhibits the COX enzyme, which stimulates inflammation and pain, and thus aspirin inhibits inflammation and reduces pain. So that is the majority of the enzyme topic for A-level biology, covered in a relatively concise manner. Now, there are other aspects that we can go into in more depth for different specifications, and exam technique for the enzymes topic is vital. If you're interested in covering any specific regions or you have any direct questions, I do offer tuition sessions. You can book those either by leaving a comment or heading over to my website and emailing me or filling in the form on the website. I will get back to you as soon as possible. In one-to-one sessions, we can cover exam technique using an exam technique aid that I've constructed from over 10,000 marks worth of past paper questions. We can go over your exact queries and any issues specific to you, as opposed to kind of this broad brush technique over YouTube. And also on my website, there are a ton of my free notes. And if you're applying for medicine specifically, I've got interview preparation kind of tools and notes on there. And I also offer interview preparation sessions if you're still waiting for any of your interviews. So yeah, head over to the website. It's linked in my description, Dr-Tutor.co.uk. And if you're interested or you have any crucial questions, then feel free to book a tuition session, and we can go over all of that in a one-to-one session. But yeah, I mean, that's it for today's lesson. If you have any broad questions or anything related directly to what I've said, feel free to leave it in the comments. And yeah, I hope you found this lesson useful, and hopefully see you next week. Now, very quickly, if you stayed all the way to this point, this graph is something that you can be questioned on, and we would go through this type of thing in far more detail in a lesson. But for those of you that made it to this point, try and leave a comment describing and explaining why the graph appears the way it does, and I'll try and reply to your comment, correcting you or making comment on your exam technique or your use of specific key terminology. So that's kind of like the challenge for this week.